The process of protein synthesis occurs by two steps: transcription and translation. During the first step of protein synthesis, which is transcription, a copy of the gene made from what we call RNA, the messenger RNA, is made, and it carries the information to the cytoplasm. We know in the DNA we have the sense strand, which runs from 5' to 3', and the antisense strand, which runs from 3' to 5'. The transcription of DNA into mRNA takes place inside of the nucleus. So we're going to look at how the DNA is transcribed into mRNA inside the nucleus. Every gene which codes for a protein has three regions, and they occur in the following order. The first region is the promoter. We have an enzyme called RNA polymerase. It unwinds the two strands of the double helix by breaking hydrogen bonds between the nucleotides, and it recognizes the first sequence of the gene called the promoter. It binds to it, and this is where it starts transcription. The RNA polymerase is actually the enzyme for transcription. After the promoter, we have the coding region, which is the section of DNA that actually codes for the protein, and this is where transcription takes place. We have three nucleotides inside of the nucleus, and these are going to bind to the antisense strand of DNA by the law of complementary base pairing in order to create a strand of RNA, which is going to be the messenger RNA, which is going to leave the nucleus and undergo translation into a protein. So what are the differences between DNA and RNA? First of all, RNA is made up of a single strand of nucleotides, while DNA is made up of two. Second, in DNA, the sugar in the nucleotides is deoxyribose, and in RNA, the sugar is ribose. That's actually where it gets its name from, ribonucleic acid. And the third difference is that the four base pairs, the four nitrogenous bases of DNA, are adenine, guanine, cytosine, and thymine. In RNA, there are adenine, guanine, cytosine, and uracil. So instead of thymine, instead of T, we have uracil, U. So free nucleotides will go and bind to the antisense strand of the DNA by the law of complementation. A will bind to T, U will bind to A, C will bind to G, and G will bind to C. So the strand of RNA that is formed is going to be complementary to the antisense strand. And because it's complementary to the antisense strand, it is identical to the sense strand, because the sense strand is complementary to the antisense strand, except for the few differences that we just talked about. Now, the third region of the gene is the termination sequence. It is the sequence after the coding region that tells the RNA polymerase to stop transcription. Transcription is going to end here, and it is on the antisense strand. So we see that the RNA polymerase is transcribing. We have the mRNA strand being formed complementary to the antisense strand, as we see here, until we reach the termination sequence, which is the specific DNA sequence that says stop. Transcription is over. Once that happens, the mRNA strand that formed complementary to the DNA on the antisense strand dissociates, it leaves the DNA, and it leaves the nucleus into the cytoplasm, where it is going to undergo translation. So the DNA strand that was just transcribed closes again, it becomes a double helix again, it doesn't stay open, and transcription is over.